Our today's topic is intestinal juice, sucrase entericus, also called sucrase entericus. We will be learning composition of intestinal juice, its function, mechanism of formation, and regulation of secretion. So before that, we will just go through functional anatomy of small intestine. Then the rest part will be discussed. So we know small intestine is having three parts. Initial part is after the stomach. So what is the initial part? Duodenum. It is about 25 centimeter long and almost C-shaped. And thereafter, next part will be jejunum. It is about 1 meter in length. And then ileum. So it is about 2 meter in length. Now, duodenum, duo means 12. 12 fingers width. It is almost 25 centimeters. So from that, the name derived, 12 fingers. If we go through the small intestine, it is the intestinal part, and we have taken a section longitudinally. So there will be two halves. So if you look into the inner surface, what you will find? These sort of bands. These are folded. See the bands. These are the bands. What is the purpose? Formation of these bands, round bands? Increasing the surface area. And you will find this surface area is further increased by the epithelial cells, and these epithelial cells are having brush border. So this brush border further increased the surface area. Why it is necessary to increase the surface area? Because the intestine will be performing most of the digestive functions and most of the absorptions. That's why it is long. And having increased surface area, so that during this period, during the passage of food along the length, it can be digested. So during the passage, what will be there? There will be movement of the intestine, different type of movements will be there. So because of these movements, what will be there? This will be proceeding forward, and there will be mixing of the food bolus with the juice. So this action of the intestine, mechanical function, this is one of the mechanical function of the small intestine. So with this function, it will facilitate mixing, digestion, and absorption. So this is the structural change it made to facilitate digestion, absorption purpose. Okay. And you see, these are what What are these? These projections? Villi. And these villi are lined by epithelium. These are all secretory epithelium. They will secrete something. They will secrete enzymes. They will secrete mucus. And these cells will also absorb food. For this purpose, each cell is having brush border, microvilli. Okay? And you know, these cells are acting as holocrine organs. Holocrine. What is holocrine? Every person has a particular smell. From where this smell is coming? Is it sweat? It's sebum. We have sebaceous gland. And this makes our skin oily. And this sebaceous gland, they produce sebum. And you know, each cell, the cells which produce sebum, they die. Along with this secretion, this dead cell also coming. So they will be producing secretion at the same time, along with the secretion, the debris of the cell will be coming. Here also, the apical cells, the intestinal cell lining, these cells are also not permanent. Always they are shedding. New cells are coming, taking their place. And we learned this during apoptosis. In our apoptosis class, I have mentioned that. What will happen after they will be dislodged from the basement membrane? Epithelial cells always rest on basement membrane. When they are dislodged, they are dislocated and separated from the surrounding cells. So what will happen to them? That will undergo apoptosis. Okay. So here also their place. So along with the secretions, enzymes, the whole cell will also be secreted. So in this secretion, because of this, you will find dead epithelial cells. If you analyze these sucrase entericus, you will find there are some dead cells. So you know, whatever, they will not be permanently there. So gradually, they will be shedded off and new cells will be coming from these sides. So gradually they are moving. When they come here, they will shed off. Clear? Okay. And you see, these are the villi, no? So in between two villi, see there is some invaginated portion. These. Actually, these enlarged. These are crypt of liver cone. These are crypts of liver cone. There will be these crypt of liver cone, this epithelial lining, there will be different types of epithelial cells. And they will be performing Some are goblet cells, motu cells. They will secrete mucin, mucus. What is the purpose of this mucus? This mucus will be mixed with the food bolus, so it makes this slippery so that easily it can pass through the lumen. And the wall of intestinal wall remains protected. It will not cause any damage to the wall because this will be contracting, no? This way. There will be, next I will discuss what are the different types of movements the GI tract has to perform because of it is having muscular coat, no? So with this muscular coat, they will be performing, except in stomach. In other part, there will be two muscular coat. One is circularly arranged and other they are longitudinally. And in stomach wall, there will be an obliquely arranged. So what are these cells? Stem cells. You will find there are stem cells. And we know stem cells are cells. They can be transformed into any kind of, they can produce any kind. We have seen this in bone marrow. There are bone stem cells, so they can give rise to, if necessary, WBCs, if necessary, RBCs, if necessary, thrombocytes. Okay, so they can be transformed to any, depending on the situation, requirement, they can be transformed into any other cells. So that's why the stem cells, why their need is there? Because as I said, new cells has to be produced, and because at the apical portion, these cells are dying. They are coming along with the secretions. So that has to be replaced. So stem cells are producing this, so these are coming at the T gradule one by one. What is this? Paneth cells, goblet cells, and enterochromaffin cells or argentaffin cells. So different kinds, I cells, S cells. I cells secrete. I cells, there will be I cells, it has not been shown. So I cells, they are responsible for cholecystokinin and pancreozymin secretion. Cholecystokinin has got, it is a local hormone. Pancreozymin, it is a local hormone. Cholecystokinin, actually previously it was thought, there are two separate hormones. One is cholecystokinin and another is pancreozymin. Cholecystokinin function is to contract gallbladder and to increase bile secretion from the liver. Gallbladder is not making any bile, it is just storing. So if I want to stimulate the formation of bile, then I have to stimulate hepatocytes. Okay, so it will produce bile. When it is needed, when I take lot of fat, because this fat digestion and fat absorption requires bile salts. Not only these, the bile will be having basic alkaline secretions. They are having alkaline, means bicarbonate ions. So these bicarbonate ions are also necessary. Why? To neutralize the hydrochloric acid coming through the food from stomach. When it is seen that whenever food is coming, entering into the stomach, immediately liver is prepared. Secretin, liberated. There will be gastrin, these will be liberated, no? because of the presence of food in the stomach and because of psychic stimuli. And now, when you are in a queue, before that he was there, for he will be coming for Bhaivavusi. So when he entered, immediately, oh, it's my turn. He started. Okay. So before Bhaivavusi, appearing Bhaivavusi, before that he started, he became nervous. Similarly, when we eat, immediately we think about, or we are masticating the food, or presence of food in the stomach, my later part is ready with secretions. So who will initiate this? Through nervous system and through secretion of hormones, local hormones. So the bile is ready, gallbladder is ready. When food will be entering, or before that, actually secretion starts. And when food bolus will be entering and coming in touch of the alveolar the epithelium, intestinal epithelium. So when they will contact, the food bolus will make contact with the epithelium, immediately, reflexly, and there will be secretions. If it is gallbladder, there will be secretion. If it is intestinal juice is required, there will be secretion in intestinal juice through reflex path, through reflex mechanism. And they will also secrete the local hormones from these cells. And these hormones will also stimulate. All these cells are having receptors, so they will bind the receptors and will modify the activity of the cells. So its function is to, say for goblet cell, its function is to secrete mucin, so it will secrete mucin. Another cell, the function is to liberate bicarbonate ion, so it will liberate bicarbonate. Another function of the apical epithelium, their function is to produce enzymes, so they will produce enzymes. So this way, local hormones and nervous system helps in digestion process. Okay, helps in secretion of local hormones, helps in neurally it helps in secretion of different juice. So these are the cells, Paneth cells. They secrete what? Lysozyme. They are also able to phagocytosis, and they also secrete one local hormone, and this local hormone is called, it gives, I am telling you the name, it gives protection. It's one kind of immunoglobulin. So immunoglobulins are always protective in nature. It protects our body. So this will help the protection of the epithelium, so that invaders cannot destroy, make any inflammation on the walls. What are these? These are the villi, and these are crypt of liver cone. And see, below this layer, these are mucosal layer. Okay, mucosal layer, and this muscularis mucosa, muscle in between. Muscular, this is purely smooth muscle, muscular coat, and this is submucosa, and within the submucosa you see what are these round shape? What are these? These are Brunner's gland. What? Brunner's gland, tubular structure. So when we cut it, we will get ring-like thing. So these are actually Brunner's gland. What is their function? Their function is mainly to secrete mucin. And this mucin from this gland will mainly mucin. So their secretion are also under the control of vagus nerve. So when vagus nerve is stimulated, their secretion will be increased. So there will be mucus in the succus entericus. So these are the Brunner's gland. These are what? Brush border epithelium. This cell one of the cell of brushwater epithelium. What is this? This is one cell, it is one cell. Can anybody tell? You will face one, you will find some questions from here. Not from here, I mean, these are cell junctions. There are different kind of cell junctions. Can anybody tell me what are these different kinds? One is tight junction. Okay. So you study well. Some question will be there from these junctions. Gap junction. What is the function of tight junctions? What are the functions of gap junctions? Adherence. So these things are there. In cardiac muscle, heart muscle, it is having one unique property. What is this? This is all or none law. It follows all or none law. What is that law? See, a lot of chairs lying side by side. And these chairs are myocytes, muscle cells. They are lying closely. If I stimulate one chair, at a time all other chairs start to contract. How? I have stimulated this one only. Because whatever change I made in this through stimulation, this change is transmitted to these, to these, to these, to these, to these, to all the cardiac muscles. As a result, when it is excited, all the muscles will be excited. When it is contracted, all these muscles will be contracted simultaneously. No need of stimulating each one of these cells. That when the stimulation comes to a threshold level, then only this cell will be stimulated. Before that, will not be stimulated. I'll take another class on action potential. I'm not getting time. So, action potential, we have learned how resting membrane potential is generated, and next part was how action potential is generated. That is the most important part. This will be coming repeatedly while we will be discussing the nervous system. We will be discussing the nerve muscle physiology. So this is important. Okay. So when stimulation, not every stimulation can create action potential. It should have a sufficient strength capable of producing action potential. And this value, see, when you are asleep, so if somebody touches you, you are not able to feel. But when the strength of the stimulus is much more, then now it is having the threshold stimulus. It has crossed the threshold stimulus. Now it has initiated action potential. So when a stimulus capable of producing action potential, that is called, that strength of the stimulus is called threshold stimulus. So every stimulus to produce effect should have a threshold stimulus. If it is sub-threshold, sorry, I will not respond. And if it is more than the threshold, same result. 5 electric current of 5 millivolt, it is threshold. 6 millivolt, 7 millivolt, same effect will be there. Because I have attained the threshold and I am now capable of producing action potential. Okay, since action potential has been generated, now we know these are propagatory in nature. This action potential travels along the sarcolemma, along the axon. If there is along the axon, if there is Schwann cell, so there will be node of Ranvier. So this action potential will jump from one node to another node, one node to another, and this type of conduction we call saltatory conduction. Can you remember this? Saltatory conduction. Okay. So if there is no myelin sheath, the conduction will be like this. Gradually it's coming, but its speed is less. But when saltatory conduction will be there, we will be traveling like this. In between, these are non-conductor of electricity, myelin sheath. So there will be jumping. So velocity of nerve impulse will be higher in myelin sheath. In those nerves which are having myelin sheath, modulated nerve, we call them modulated nerve fiber, isn't it? Okay. This is also important. And it will be discussed during absorption of food. So now I'll take 1 minute for this. See, these are what? Villi. This is one of the villi. And these cells are having microvilli. It is not been shown. So they are absorbing food. These cells are absorbing food. So now, what they will do? I am the cell, I have absorbed the food from the lumen. Then what to do? I have to send it to the blood. No? But there are some products, digested products, which are big. Say, for example, long chain fatty acids. Say, for example, monoglycerides, diglycerides. What will happen to them? These are big, no? So there will not be further digestion. So that has to come into the blood. Now how they will be coming? This is a branch of artery entering from this into the villi and coming back to liver. From the liver, it is entering the heart. And when heart will be pumping, it will be circulated throughout the body. Here, those who are diffusible, the substance which are diffusible, they will only enter into the blood. Because the concentration of those substances are more in the cell and concentration of those particular substances is less in the blood. So there will be concentration difference. So because of concentration from higher, within the cell it is high, but in the blood it is less, so there will be diffusion. So by simple diffusion, these substances will enter. But as I said, what will happen to the bigger molecules? Bigger molecules will travel through another path. This is the path. What are these? This duct is called, the green colored duct is called lacteal. You have heard the name of lymph. These lacteal ducts actually contain lymph. These are white in color, not red, because no blood cells are there. Blood cell cannot enter there. Okay? So, the big size molecules As I said, the name, what are the examples of? Monoglyceride, long chain fatty acids. Okay, they will enter into the lacteal, they will form one thing, chylomicron. What? So within the chylomicron, see, the outside, some molecules help. They help the formation of chylomicrons. One is bile salt. Bile salt reduces surface area, we know that. So bile salt will help in the formation of chylomicron. So their molecules will be arranged circularly. And they are hydrophilic. Means outside now became soluble in water. And within this, there is a pocket. And in this pocket, what will be there? The big size molecules. They will be inside. Okay, so outside these substances are water insoluble, hydrophobic, but I have made hydrophilic layer. So this type of particle, we call it chylomicron. So what is there inside? There are those big size, relatively bigger size digested products. Outside there are molecules of bile salts. So with this, they will pass through the membrane because it is hydrophilic. The outside is hydrophilic. So they will pass the cell. So they will pass the basement membrane, the cell membrane, and will enter into there in the form of chylomicrons. Now they will pass through this. This is now lymph, through this lymphatic vessel, lymphatic vessel, lymphatic vessel, lymphatic vessel. This is green one. Now this is what? Left subclavian vein. So they open into the subclavian vein. And this will end into superior vena cava. From the upper part of my body, venous blood is coming through superior vena cava and will drain its blood into which part of the heart? Atrium, obviously. Which atrium? Haji, you must learn this. Circulation of blood through vessels and heart. The venous blood of upper part is coming through superior vena cavas. And venous blood from the lower part of the cavity, lower part of the body coming through inferior vena cava. Superior, inferior vena cava. And both of them will combine and will drain their blood into right atrium. Okay? From the right atrium, it will enter into right ventricle. And when the right ventricle will be filled up, it will pump, it will contract, and this blood will be coming to the lungs. Why it is coming to lungs? Because all this venous blood is having less oxygen, more carbon dioxide. So it needs oxygenation, and carbon dioxide has to be eliminated. So in the lung, while passing through the lung, it will attain oxygen, leave carbon dioxide. Then it will be back to left atrium. Now from left atrium, left ventricle, now the left ventricle will contract and this blood will be circulated throughout the body. Okay, since this vessel is draining the lymph into what? Subclavian vein. So this subclavian vein will be joining the superior vena cava So from superior vena cava it is coming to heart. From heart it again after passing through the lung, then it will be circulated through arterial blood, and then through hepatic artery it will be coming to liver. Then, okay, these are the things I have absorbed from intestine. Now whatever you want to do with this. Understand this? Okay. 99.5% is water and only 0.5% is solid. So among these solids, some part is organic and some is inorganic. Here mainly what I will be getting, bicarbonate. Other anions, cations are there. And among the organic substances, we will get enzymes and we will get other organic substances. I told about in previous slide, I couldn't remember the name of the local hormone, but I said these are one kind of cytokines. Protection. It helps in protecting our body. So what are the actual name? Defensins. Defensin. See, other organic component, defensin. Intrinsic factor, we learned, intrinsic factor is produced where? Intrinsic factor? Huh? Hello? Stomach. And it is needed for vitamin B12 absorption. So our intestine also produce some intrinsic factor. They also produce some intrinsic factor. Purpose is same. Okay? And mucus we have seen, and we have already learned the functions of the mucus. Protection, easy passage of chyme, easy passage, and protecting the wall, intestinal inner wall, epithelium. And these are basic also in nature. So they protect from injurious effect of hydrochloric acid here. So these are the functions they are doing. While studying hydrochloric acid in stomach, the functions of hydrochloric acid, I forgot to tell you one thing. We have seen in stomach, no digestion of carbohydrate take place. Some part of digestion takes place because of ptyalin present in the food bolus. But after some time, 3 or 4 minutes, this activity is lost because of high pH, low pH, acidic pH. But we are taking sugar, no, with some, our cook adds some sugar in every item. Okay, almost some sugar, 1 spoon, half spoon. So they are provided. We take tea, we take sweets. So sweet is a common thing. Every day we are taking sweets. Means these sweets are what? Carbohydrate. What type of carbohydrate? Can you tell me what type of, every day you are eating carbohydrates? One is starch. One is starch through rice, through cereals you are eating. Another is cellulose. And other than this, come on. If you take milk, whether it will be having any carbohydrate? No. No. Or yes. What carbohydrate you will get? Don't know? Can you name some, in the market it is tinned, product is available for babies. Can you name them? Cerelac. And before that? Before that is lactogen. Lactogen. Lactogen means one carbohydrate is there. Lactose. Lactose. It's a disaccharide. Reducing disaccharide. It's a lactose. And lactose are having two monosaccharides. What are these? Glucose and galactose. These two are isomers. Since both are having C6H12O6, but the structure is a little bit different. Orientation of hydroxyl groups is different. Okay, so this lactose has to be digested. How? There is a specific enzyme secreted by intestinal juice. We call it lactase. So lactase will split lactose present in milk into glucose and galactose. Now they can be easily absorbed. You are taking sucrose, chini, sugar, isn't it? What are these? Disaccharide. Is it reducing or non-reducing? Huh? Some are telling it is reducing, some are it is non-reducing. Why? Because I know less, so let me tell, you can make me corrected. One is disaccharide, one is glucose, another is fructose or fructose, whatever you say. Okay? Glucose, fructose. Both are having reducing group. Both are reducing sugar when they are separate. And this glucose is having, what is the reducing group of glucose? Aldehyde group, CHO. And what is the reducing group of fructose? Keto group, keto, second carbon. If I ask for a name one keto sugar, keto monosugar, monosaccharide, what will be your answer? Fructose. And keto disaccharide, this disaccharide will be having keto group, may not be free. Chini, sugar, sucrose. Aldehyde group at carbon 1 and fructose, second carbon is keto group. So there will be formation of glycosidic linkage and it is in between 1 and 2 keto group and aldehyde group. Both will form the glycosidic linkage, 1,2 glycosidic linkage. As a result, both the reducing group lost their reducing ability. That's why they became non-reducing. Am I correct? More than you are expert now in biochemistry. Okay. So we learned the function of sucrase. Sucrase splits sucrose into glucose and fructose. Lactase splits lactose present in milk into glucose and galactose. And maltose. Do you eat malt?
Yes.
Definitely you take biscuits, bread. These are fermented. Okay? Malt is produced. So there will be some maltose. Maltose is also a disaccharide and these are non-reducing, reducing disaccharide. Reducing disaccharide. So if you split maltose, you will get two molecules of two monosaccharides and both are glucose and glucose. When they are linked together by 1,4 glycosidic linkage. So first glucose, its aldehyde group is connecting to hydroxyl group of fourth carbon. Fourth carbon. So its first carbon aldehyde group is free. That's why this disaccharide is reducing. One is free. This one is used in the bond formation. This is free. That's why it is reducing. So, maltase hydrolyzes the 1,4-glycosidic linkage of maltose. As a result, two molecules of two monosaccharides are formed, glucose plus. See another, have you ever heard the name trehalose? This is also a carbohydrate, disaccharide, having glucose. But these are fly. Some fly while they travel to a distant place. So from where they are getting their energy? From trehalose. And while they are travel, there will not be any water. So what is this trehalose? It absorbs some water. So it provides necessary water as well as necessary energy. For flying, the energy will be needed, no? Some insects we relish. See, go to China, you will find. Go to China. So different types of, starting from cockroach to anything. They eat it. They fry it, eat it. Dry fry even. So these insects are also having trehalose. So that is not known by our GI tract. So it will secrete trehalose, this enzyme, trehalase. So if any trehalose is there, some vegetables also are there which contains trehalose. So this has to be digested by trehalase. So after this digestion, that we will get two glucose molecules. Dextrinase, we have already discussed that after salivary digestion by the ptyalin, the product is dextrin, maltose, maltotriose. So these dextrin are having lot of 1,6-glycosidic linkages. 1,6-glycosidic. That's why ptyalin, or if it is it is alpha amylose, or salivary amylose, or it is pancreatic amylose, they cannot break 1,6-glycosidic linkage. So that part of undigested carbohydrate is called dextrin. These dextrin are digested by dextrinase into glucose disaccharides. So you will find some disaccharides, you will find some trisaccharides. Any compound having 3 glucose molecules, trisaccharides will split this, and ultimately you will get 3 monosaccharides. Disaccharides, example is maltase, sucrase, lactase. These are disaccharides. Okay, so these are the functions of these enzymes. Now you see in the proteolytic enzyme there are peptidases. From the digestion of trypsin, from the digestion of chymotrypsin, from the digestion by pepsin, we are getting lot of peptides, peptones. These peptide and peptones will be digested by peptidases. So suppose these are the peptidases, peptides. So these peptidases, they will break these bonds. As a result, dipeptide, tripeptides will be formed. There are dipeptidases, means 2 amino acids, they will break this, so we will get 2 amino acids. There are tripeptidases, they will break the 2 peptide bonds in between the 3 amino acids. So in this way, the proteins will be digested. And you know the function of the, and another is the enterokinase. Do you know the functions of endokinase? Activation of trypsinogen into trypsin. So with this, we came to know the functions of all the components of intestinal juice. Okay?